This video looks at multicellular life, and it's part of the evolution of organisms topic. In particular, we're going to be looking at what are the challenges faced by the evolution from single-celled life towards multicellular life. Some of the concepts from the subject outline include that organisms are going to survive most effectively within their tolerance limits. And so things like temperature, water availability, glucose, carbon dioxide need to stay within those limits. And how do they face those challenges and how do they overcome them? When conditions change, how do they return to those environments? And in order to do that, they need to detect and respond to those changes. How do they do that? And then the concept of homeostasis and the requirements of that. So those are some of the key parts of this topic we'll explore in the video today. So I want you to begin by thinking about a cell in a tissue. So I've got here a picture of the urinary tract. In particular, let's look at a cell in the organ, the kidney. So this cell is deep within your body. In a single-celled organism, that cell is in close contact to the environment. So it's got oxygen right there, it's got water right there, it's got nutrients right there that it can take in. And if it's got wastes, it can expel those directly outside of the cell and they go into the environment. But a cell that's deep inside your body doesn't have access directly to water or oxygen or nutrients. It's surrounded by other cells. So if we have a look at this cell, and perhaps we have a look at a particular cell here, this cell here isn't in contact with the environment in any way. So how does it get water, oxygen, nutrients? How does it get rid of wastes? Well, the clue for that is right next to it. So even though the cell is surrounded by other cells, it's pretty close to a blood vessel over here. And in between those cells is extracellular fluid, or fluid outside the cells that the tissue has. And so anything that's in the blood can diffuse through that fluid and reach its way here. So if there's oxygen here, the cell can get it by diffusion across that gap and through that fluid. If there's waste, they can diffuse towards the blood vessel. So let's have a look at how this works in practice. So the challenge for multicellular organisms is that their cells aren't in contact with the environment. And the solution to that is to kind of bring the environment to them. And circulation in us and other multicellular organisms is a really good way to do that. So here we've got circulation happening through the circulatory system, the blood system. The red blood cells here carry oxygen inside those cells. And so by bringing the oxygen from the air, using the lungs and the red blood cells to the cells in the body, oxygen can diffuse to those. And the reverse happens. The cells are producing carbon dioxide as a result of cellular respiration. That needs to go somewhere. That will diffuse into this tissue fluid and diffuse to those red blood cells, which are now heading back to the lungs to get rid of that CO2. As well as the cells in the blood, obviously there's fluid here, the plasma fluid. In that there'll be glucose. Where does the glucose come from? So the glucose comes from the digestive system. So when you digest food, it's broken down into small molecules like glucose. The glucose goes into the blood plasma and again will diffuse, might require some facilitated diffusion, but in the fluid it can diffuse through there and enter cells through either facilitated diffusion or active transport. So again, you're bringing the nutrients to those cells that are deep in the tissues. So as we can see, different organ systems, the circulatory system with its vessels and the heart to pump that around, the digestive system, the respiratory system, each of these are able to bring that sort of environment to the cell so that even in a multicellular organism, the nutrients, the gases, the wastes, they can all be exchanged with the environment and either brought in or disposed of. So the other things that are about the tolerance limits, we can think of about sort of temperature and water availability and things like that. So a multicellular organism has a number of different things that it needs to maintain in order to keep those cells healthy, because there is a difference between that external and internal environment. For our low-res rabbit here, the external environment might be 20 degrees with a very variable chemical composition and a high degree of entropy, which means that the level of disorder, the stuff that's out in the environment is pretty disordered. Whereas inside the cell, we have highly ordered, highly structured molecules like DNA or proteins. The internal environment is nice and warm, the chemical composition should remain fairly constant, and the level of entropy is low. So there's a big difference between the external and the internal environment. And homeostasis is about maintaining that difference because things inside the body need to stay the same. And when we say inside the body, we're really talking about inside the cells. Because remember, cells are the units of structure and function. So the functions are happening inside the cells. They need to have all of these different things maintained in order to function properly. So how does that happen? Well, the process is called homeostasis. It's about homeo meaning the same and stasis, I guess, being like status. The situation remains the same for those cells because as we looked at with our different pracs and things like that, enzymes work best at a certain temperature. You want to maintain temperature. Reactions will depend on the rate of reaction being controlled by the substrate concentration or the product concentration. So again, those need to stay fairly constant as well. So here's an example of how homeostasis happens. So homeostasis we can think of as this balance. The seesaw is perfectly balanced. If an imbalance is caused by a stimulus that's tipping that up, for instance, that change needs to be detected by a receptor. This sends an afferent or an ingoing pathway to some sort of control center like the brain. There's an output from an efferent pathway going out from the brain to an effector. The effector is something that can cause a change to happen, a response, and that will tend to push up on this side, for instance, and maintain that balance. So homeostasis doesn't actually keep things constant. What it means is that there are these tolerance limits within healthy or well-functioning ranges for cells, and any time that they start to get outside of that healthy range, then homeostasis can tip that back. Those mechanisms can tip that back into being within the control range. So whether that's temperature or whether that's the amount of glucose, those are maintained within those tolerance limits. So how does that actually work? Let's break that down a little bit. So you need to have a receptor that can detect a change, whether that's a change in the external environment, such as the temperature, or the internal environment, such as the amount of glucose in the blood. Something needs to pick that up. Then there needs to be some sort of processing information. So detect this change, what are we going to do about it? Send a signal, and then other tissues, the effectors, can cause a response. So some sort of receptors might include sensory neurons, and we're aware of those external sensory neurons, or the ones that detect the external changes, taste, touch, sound, sight, smell. There's also ones that detect internal changes like blood pressure, body temperature, hunger, thirst, and balance, our sense of balance, and those require those internal receptors as well. So there's a whole suite of different receptors and sensory neurons in your body to detect those changes. That information is then sent to different places. So depending on what kind of information is coming in and how quickly the response needs to happen, that might just go to the spinal cord, just the spinal cord and then back out again for a quick thing like a reflex. Some unconscious things only get sent to the lower parts of the brainstem or the cerebellum. So thinking about breathing or hunger, thirst, balance, things like that, they're controlled at an unconscious level. And then other things such as your sight and sound, they're sent to the brain for further processing and complex decoding. Wherever the information goes, there's a process and thinking about how we're going to respond to this change. And that's done by effectors. Probably the easiest thing to think of is the muscles. So skeletal muscle will change or respond. So for instance, in terms of your balance, you might need to tense or relax different muscles. Other things like, for instance, sight, if you're getting too much light and your iris needs to contract, that's also a muscle, but it's a type of muscle called smooth muscle. Moving food through your digestive system is also a smooth muscle. And then there are certain tissues like glands where a response is received and they will send out hormones or other signaling like that in order to have further downstream responses. And that's the sort of system that's used in glucose metabolism. So you can see that, again, the simple thing is a receptor receives the information, it gets processed, and then effector causes a response. We tend to then divide these loops, stimulus response loops, into two main types. The first is called negative feedback. Negative feedback is caused because whatever the response is has a negative effect on the stimulus and tends to oppose it. So for instance, body temperature is a really easy one to look at. So here's body temperature within the normal range, which is what it should be. If the body temperature is too low, then the effectors might use heating mechanisms such as shivering. So your muscles are undergoing metabolic processes to create heat. That heat then increases body temperature, and so whereas the environment made it go cold, the shivering warms you up again and opposes that and brings you back to that normal range. On the other hand, if you're too hot, then there's other mechanisms such as cooling mechanisms. So again, sweat gland might be producing sweat in order to provide evaporative cooling and cool you down. That body temperature decrease then opposes this one and brings you back to the normal range. So most of these homeostatic mechanisms are about opposing the change to bring you back within the tolerance limits, and that's negative feedback. There are some examples of positive feedback though. So positive feedback is where the stimulus then creates a further stimulus that makes a change in the positive direction that feeds back and keeps building and building like a chain reaction. In this case, you're not trying to keep things the same, you're actually trying to cause a change of state. And the classic example of this is around pregnancy and birth. So when the womb is fully taken up, the uterus is fully taken up with the baby and time for birth is about to happen, and that's going to put a stretch onto that uterus that will cause a positive signal for release of a hormone, oxytocin. This causes the uterine muscles to contract, and obviously when the muscles are contracted around the baby, that makes it feel even tighter and stimulate those stretch receptors more. And so you have a positive feedback loop going round and around here, leading to further and further and stronger contractions, until the contractions get so strong that it's time for birth and the baby is delivered. And of course, once that happens, then suddenly the state changes and you no longer have the activation of the stretch receptors. None of these positive feedback loops can go on indefinitely, otherwise they will just build up. So somewhere on the line, there has to be a change that happens. And so those are negative feedbacks and positive feedback loops.